first encourage all the students here to please let me know if there's something that you are not understanding. I have tried to keep it extremely basic, tried to keep it my talk steered towards undergraduate audience here, and so therefore I would really love to have the participation of the students. Um, so to begin with, I just want to say that I work, like Dr. Chakrabarti already introduced, I work on sound using various different model systems, and some of the model systems I have worked on, I have put it on the slides. I have worked on moths, I have worked on birds, bats, and insects, like nocturnal insects like crickets, and of course moths, like I said. I've also worked on primates and fishes, but not really using sound in case of fishes. So basically I'm broadly interested in the diversity of life that we see in and around us. And I am thrilled that some of us are so lucky to be born in a tropical country where there is biodiversity in our backyards. So, having worked in the Western Ghats, in the rainforests of the Western Ghats, the dry deciduous forests of Western Ghats, scrub forests in Western Ghats before I joined ISER Mohali, and now working in the Himalayas and in the urban landscape, I find that there's an incredible variety of organisms that we are surrounded by, and I am fascinated by this diversity in life. So, this is what largely I work on. But I also look at not just patterns of diversity, but I'm also interested in the behavior of various animals, and which includes nocturnal insects, this cricket, which produce a lot of sound, but also birds. So now my major focus is mostly on birds and on cricket. This is my lab group. These are my students and the bunch of really fun-loving people. We are all interested in this diversity of life. We all work in both in the lab and in the field, but primarily in the field. And Indian Institute of Science, Education and Research, Mohali, is in the foothills of the Himalayas, and this is exactly where it is. So just a 1 hour car drive will take you into the Himalayas. So I'm very lucky to be situated in such a nice location. The major focus of my work, just to give you a very quick introduction for students who may be interested, I work on sexual selection, sensory ecology, social behavior, but for all of these things I use bioacoustics as a tool to study various aspects of different kinds of model systems, whether it is birds, whether it is fishes, whether it is insects. And with that short introduction, I would dive right in. Since I was told the workshop is on understanding methods in faunal surveys in ecological studies, I have tried to, in my talk, I've tried to give you an overview of the traditional methods. Being a field ecologist, I would like you all to get a feel for what you actually measure, from where this data comes, which actually goes into testing some of the theoretical models that Professor Ray talked about. So, getting this data is a non trivial task. It takes a lot of time to collect data from the field, and oftentimes years of work will collapse into one graph. So, that is something that I want you to bear in mind, that that is the occupational hazard that we have. Now, ecological studies, most ecological studies depend on measuring species presence absence. So, then from there, richness and abundance of different species. And for this, we have to rely on various methods in ecology. Now, this can be done both for plants and for animals. I specifically work on animals, so that is what I'll talk about. And this is what Zoological Society Kolkata's workshop is also on, faunal survey. But the choice of the method will really depend upon which species you want to work on, or it could be the taxa that you want to work on. You're working on insects versus you want to work on mammals. You're working on a nocturnal species or a diurnal species. So it really depends upon that. It also depends upon the terrain in which you have to collect data. It also depends upon the kind of question you're really asking. So the choice of method really depends on each of these. how much data can you collect in the given short period of sampling that we have now? In ecological time scale, 5 years of PhD is also a short period, right? So from that perspective, the resolution of data, the main constraint is how much time do you have and how much effort can you put in collecting this data. So these are the constraints, okay? And another thing I want to keep I want you all to keep in mind is that the traditional methods which I will overview, many of them are actually destructive, or these are invasive sampling where you collect the specimen, which means the specimen, the individual is um, basically you collect it and it is fatal, so you kill the animal in the process. But it is reliable because you have the specimen and you can identify, you can do all kinds of other interesting things. So it is destructive and invasive, but it is reliable. The question that many of us faced as we were doing our PhDs was, can we do this without being invasive and yet still being reliable in our data? So that is where I'm coming from, and that is the idea behind introducing you to this contemporary technique of bioacoustics, this tool that we use, bioacoustics. And I would like to make a case here that why bioacoustics is a reliable, non invasive technique to study patterns of biodiversity. I have been studying various species using bioacoustics since 2004. It's been a very long time now. And I am convinced now that this is definitely a very powerful tool, and more it is going to become cheaper to use sound to study our ecology. So now when we talk about ecological sampling, the first thing we talk about is whether we can get at abundance and density. And to get at abundance and density estimates, there are two factors: the size of the animal and the mobility of the animal, which will determine which method we will use. If it is very small, for instance, if you are sampling beetles or moths versus if you are sampling elephants or tigers, and whether you are studying barnacles, which are basically more or less sessile, versus if you are studying whales. Okay, so size and mobility are two factors that you need to keep in mind, and the methods can either be an estimate or actual total count. So from this dichotomy, let's look at the various methods, the traditional methods which are used as total counts or censusing. So, where you count all individuals to get at abundance or diversity estimates. For example, the human census. When faunal survey, then we mostly talk about non-human animals, but the human census is one of the most spectacular way in which data is collected about how many individuals are there in a given area. So it's a fantastic example. Camera traps that are used to get at total number of individuals in a given area. There are acoustic surveys. Then indirect methods of total count, we use pug mark analysis, scat or dung analysis. These are what we call as total counts, where you're counting actual numbers. On the other hand, you can also get estimates from sample of the population is taken to get an understanding of the relative abundance of one species as compared to some other. So there we use quadrat techniques, point counts, transects, mark recapture. These are estimates. So very quick overview of these traditional techniques before I go into the contemporary ones. The human census, as you know, you all have at some stage been part of it, where in people actually go and survey from door to door, counting exactly how many. And this has been a fantastic exercise in India, religiously carried out, and it's a great way of doing total counts. But can we do census for animals? Yes, we can do census for animals where we can count exact number of individuals, but it depends when and where you are going to do and for which model system. For instance, in the Serengeti to Masai Mara migration of the blue wildebeest, if you time your census at the time of the migration, it is very likely you are going to get a very close actual value of the total count of these individuals. So you have to time your census, and whether the species is a species that shows a certain kind of behavior. So you can exploit the behavior of the species to design your survey. On the other hand, there are some species who are not migrating, but then they gather together for certain kinds of uh, uh, in certain life stages. For instance, during breeding time, they may gather together in large colonies. Here again, as you can see in this case, these penguins, it is possible to actually do total counts of the species, or when you count barnacles, where in any species where they gather together either for a particular life stage or for a particular time of the year. Here in the Christmas Island, red crab migration happens at a certain time of the year, and in fact they close the roads to vehicles because they are allowing the crabs to migrate. Here again, census, total count is possible. So I'm just trying to give you a sense of the method, but this method can be applied for some species. It may not or cannot be applied for certain other species. It is not that easy to apply it for some other species. Tigers, for instance, if you want to get total count, back in the day, individual the forest department would actually run transects, and total count would happen by manual surveys. Now, of course, we know camera traps do a good job of monitoring dangerous animals, nocturnal animals, or shy animals, or those animals that have very low population density. Um, and the advantage of using camera trap is that you can monitor these animals in their habitat without actually disturbing them, without you physically being there. So it is truly non-invasive method. You are not even physically present. Of course, you are not killing the animal in um, counting them, but you are also not even present there. So this is truly non-invasive, and a lot of work has happened in India using camera traps. The flagship program of the, well, as you know, the Tiger Project has been running, and now more and more camera traps are being used, but also for many other animals, including the striped hyena, for wolves, for desert foxes, all kinds of animals. We are using camera traps. A little just a brief point about camera trap for the students. The height at which the trap is installed is important because that depends upon which animal you are trying to capture or trap. Okay, so in your camera, which animal are you trying to trap? If you are trying to trap a giraffe which has really long legs, okay, so the beam is not going to cut easily if the camera trap is placed too low. Okay, on the other hand, if you wanted to survey porcupine, then if it is placed too high, then you have a problem. So therefore the height of the camera needs to be positioned or adjusted depending upon which species you are trying to get. Then, of course, it also depends whether there is a transect. You have to have some information whether the animal actually uses this path or is likely to use these paths. Or you can have a even sampling design. So you can either be empowered by previous surveys, manual surveys, where you know that these are places, say for instance, a water hole where the animals are likely to visit, so you can place it there. Or you know that these are the tracks where the tigers are likely to visit, so you position it strategically. Or you can position it, if you have a lot of money, then you can actually have a grid and evenly distribute your sampling points. So you have a lot of camera traps, and then you place the camera traps evenly throughout the forest. Okay, that is another sampling design. Now, I just want to show you how this camera trap thing, like how does the analysis basically work. I'm sure most of you may know some basics. Just to give you some idea, these are two different individuals actually. And if you look at it carefully, while the patterns here, if you see on the abdomen, there are similarities, but if you look at it carefully, there are clear differences on with respect to these patterns on these two individual tigers. Okay, especially if you look at the face marking, which is one of the very important areas where the camera trap images are analyzed. You can see that these are clearly different individuals. What is important is that you should have images of animals from the same side. In many cases, the two sides, the patterns can be very different. In some other cases, the patterns on both sides can be symmetric. But here, for instance, if you have the same individual coming from left to right versus from right to left, you might count it as two different individuals. So one has to be careful. You need to know a little bit about the biology of the animal before you do these things, right? So this is something you have to take care of. Now, suppose if you know there's a species where there is asymmetry in the patterns and the stripe patterns, then you have to ensure you only use data where they're coming in the same direction. What typically is done is that the entire, if you get say just the flank, or if you get the entire abdomen, if you just get the back, the entire area is divided into a grid, okay, and every grid is then analyzed as to what pattern exactly exists, and then this is matched with other individuals. So there are softwares that do this, and which can give you an estimate of how reliable your total count is, okay. So it will match the patterns and tell you how many tigers have you trapped in your camera. Just a very easy way to identify species, of course, as we know, is using different coat patterns. So this is for a cheetah. This, you see, these are spots, okay. So here I would like you to spot the difference in the spots. So here these are just spots. Here you see this is a leopard, where you have in the leopard and the jaguar you have somewhat similar pattern of like rosettes, but here this rosette is formed of spots and then there's a central thing. In the jaguar, on the other hand, these are more, kind of merged together, and there's a central spot as well. So there are differences in the code patterns which will tell you species identity, but within a species the pattern, in case of some species, may be different across different individuals, which can give you an estimate, which can give you a good idea of the total numbers of individuals in that area. For birds, on the other hand, we use misnetting as one of the techniques for sampling how many species are there and how many individuals of the species is. Misnetting is basically where you put a net which is very fine, and from a distance, I have taken a picture from very close quarters here, so you can see my students extracting the bird from the net, and here you can actually see the net. But when you look from a little distance, the net is basically practically invisible, and then the birds, when they fly, they get trapped in the net, and there's a small pouch like thing. The net is made in a manner that there are pouches, bags that form at every level, and as the bird encounters the net, it just falls into this pouch, and all you have to do is to extract it from the pouch. However, misnetting is a very highly skilled technique, and you should not try it if you have no prior training in this, because you can actually potentially injure the bird if you don't know how to take extract it from the misnet. So all kinds of birds can be trapped. There's actually a misnet here. I'm sure you can't even see the net here. There is a misnet here from where we extracted. This is hornbill species which we extracted. And on the other hand, so it depends which bird you are trying to trap. So you will have to have the height of the misnet adjusted according to which bird you are trying to trap. And there are other intricacies in misnetting, just as in any other sampling technique. Another sampling technique of getting total counts of number of individuals is if you have territorial birds. If the bird is territorial, then for every area you can just count total number of singing individuals, and that will give you a good estimate of their uh, abundance in a given area. Indirect estimates, indirect methods would be dung and scat sampling. So you can just see, so you there are species level differences between the pellets of a cheetal versus a sambar pellet, okay. They look different, so you can just tell exactly whether they're different just by looking at the shape of the pellet. You can get an estimate of the relative abundance when you do scat sampling. But as you know, now technology has advanced. You can just extract DNA from these dungs, from the droppings, and from the DNA, whether it is of a carnivore or herbivore, it is possible to get at individual identities as well. Another traditional method that was used for a very long time for censusing and has now more or less been abandoned is the pug mark analysis. The pug mark analysis is still useful for species identification, but no longer used for individual identity. So no longer used for censusing. So here I've just shown you some different pug marks. These are not human pug marks actually. These are pug mark of a bear. This is, I think it is the sloth bear. This is the pachyderm. So elephant. This is Asian elephant pug mark. Canid versus felid per mark. You can make you can look at the pug mark style, and you can tell whether it's a canid or a felid pug mark. Cat pug marks or felid pug marks do not have claw marks on it because they have retractable claws. On the other hand, canid pug marks will have claw marks. Plus, the gap, there is always a gap between the toe pads and the heel pad in case of in case of canid pug marks, whereas in felid pug mark, this gap is missing. Okay, so these are different ways in which you can identify which species it is, but pug mark analysis, earlier it was used for individual identity, but we now know it is not reliable. Okay, so this was for larger vertebrates. Okay, but what about invertebrates? Again, we know there are many traditional insect sampling techniques. For example, the sticky tape. So you will just hang this sticky tape, so you'll get these tiny flying insects. Terrestrial crawling insects, you can do pitfall sampling. For flying, low flying insects which are present in grasslands, we use sweep nets. There are malai trap, and of course, the very destructive fogging technique. Each of these are essentially destructive sampling but reliable sampling technique from which we can get not only species identity but also estimates of their relative abundance and densities. Nocturnal insects, for instance, for moths, we do a lot of moth work. We use light traps to attract the moths, and from there you can get species identity, but you can also do relative abundance of moths in different areas. But each of these, as I said, is a destructive method or an invasive method where you are physically having to access the habitat of the animal apart from camera trap. Camera trap, where you install the device and you leave. Now, the alternative that I am proposing here is bioacoustics. Now, what is bioacoustics? As you know, sound is a means of communication in many different animals across the animal kingdom. The huge spectrum of organisms that use sound for communication, which includes insects, amphibians, birds, mammals, animals that live in water, so terrestrial, aquatic, or even arboreal animals. All of them, many animals use sound. So, sound is a good indicator of the presence of many different categories of animals. There are, of course, animals that do not produce sound, there that is there, but by and large, in every ecosystem, we will get a large number, a huge component of the community which is acoustically active, and therefore we can use sound as a proxy for the presence of the animals. So, bioacoustics is an interdisciplinary science which is in the interface of physics and biology. It typically deals with how sound is produced in biological system, how it is perceived, how it is transmitted. It also encompasses studies in the neurophysiological processes of sound production, perception, and not transmission, but sound production and perception, the anatomical basis of these, the functional significance of sound communication in animals, and its evolutionary implications. All of this comes under the larger umbrella of bioacoustics, and it's a relatively new field of study. Now, for an organism, what is the advantage of using acoustic communication? Now, if you think about what is a sound wave, it is basically a mechanical wave, okay, which is a disturbance which propagates from particle to particle interaction. So, therefore, it requires a medium. So, there are particles, the medium is composed of particles, and these particles are disturbed. Once they're disturbed, then this one particle will disturb the next one disturb the next one, as you can see. So, it is a uh, it requires a medium, and this is particle to particle interaction which is resulting in a mechanical wave. So, and it is a pressure wave which is a series of compression and rarefactions. So why compression? As one particle interact with the other particle, a compression is created, but when this wave moves forward, then there is rarefaction. Okay, so it is a mechanical wave, longitudinal wave, which is the source of the sound. Now, For an organism to use sound, what is the point? What is the advantage? Now, if you think of sound as a proxy for the presence of organism, sound source localization is very good. Just as visually, if you look at visual signal, the source localization, from where is this signal coming? Visual signals, obviously, source localization is very good, but as is for sound, okay? Visual signals, if there is an obstacle, then the sight is blocked. But sound can travel around obstacles. So, in that sense, it gives you an advantage. Sound is independent of light. So, it is good both for nocturnal and diurnal animals. And since it is a medium based communication, therefore, both in terrestrial ecosystems and in aquatic ecosystem, organisms use sound, and therefore you can use sound to survey ecosystems which are both marine or well, aquatic or terrestrial. Okay, so these are the advantages of sound for the organism, but also for the person who is surveying these animals. Okay, now, sound, a few basic principles of sound. The speed of sound changes depending upon the medium. There are factors that are going, the various factors that will affect the speed of sound: temperature, humidity, pressure. However, for all practical, and we need, since we study bioacoustics, we have to worry about all of these things. But for someone who's sensing organisms using sound, you don't need to worry about it. If you receive sound, that means that organism is present. Okay, now, just since it's a workshop and I'm introducing you to bioacoustics, it is good to know some features that we use in bioacoustic studies. All time domain features are called temporal features. All frequency domain features are called spectral features. Okay, frequency in this case, I am not talking about the count number of times. Well, the frequency of the wave comes from the cycles per second, but here I'm talking about, if you like, the pitch of the sound. The third feature of the sound that we measure, and it is important, which is used for identification and various other purposes, is the amplitude, intensity, loudness of the sound. Okay, these are three different measures of the same thing, which is the amount of energy that is present in the sound. Now, time domain, for example, here I'm showing you a cricket call. How many pulses per unit time is there? It is a time domain feature. How much silence is there between two pulses? It is a time domain feature. And how um, these chirps or these sound these groups of pulses, how they are organized, that is also a time domain feature. So in this particular case, in this is a cricket's call, multiple pulses when they group together, it is called a chirp. And this, from the start of the chirp to the end of the chirp, is called chirp duration. From the start of one chirp to the start of the next chirp is called chirp period. Same thing can go for the pulses, which we call a syllable, syllable duration, syllable. These are all time domain features. On the other hand, frequency domain would be how energy is distributed across different frequencies. And this, while time domain features are represented using an oscillogram, frequency or spectral domain features are represented in what we call as a power spectrum. Okay, and various spectral features are used for analyzing sound, which includes fundamental frequency, dominant frequency, carrier frequency, harmonics, bandwidth, various things. Okay, so there are several spectral features that can be used to analyze the sound. Now, when you visualize, when you see sound, how do we see sound? So you can see the sound either in time domain or in spectral domain, so as an oscillogram or as a power spectrum. But spectrogram is what combines both time and spectral domain features into a single plot. As you will see, on the x-axis we have time, and on the y-axis we have frequency. Okay, so this is a spectrogram, and it tells you at which time interval, under which frequency band, what kind of sound was there. Okay, so it is a snapshot of both put together, both time and frequency. It's a very, very useful, powerful plot, and this is what most bioacousticians use. Okay, spectrogram, just for you to know. Now, you can use sound, and I have used sound to understand various ecological features of insects. These are all crickets and katydids of a rainforest. Just a quick introduction to crickets. Crickets are nocturnal insects in which only males call, the females are silent. This is the wing of a female of the same species, the wing of the male of the same species, Acanthogylus asiaticus. You can see there are differences in the wing patterns. Males have specialized structures which they use to produce sound. Females lack this. Okay, and how do the males produce this sound? They produce sound by rubbing their wings together. Now, what happens when they rub their wings together? As they raise their wings together, as they raise their wings and rub the wings, the underside of the wing has this teeth-like structure, and on the edge of the wing is a hardened structure called the plectrum. So these teeth-like structure are arranged under the vein, and this is called as a file. And on the edge of another wing is the plectrum. So it is like the plectrum is scraping the teeth in this opposite direction, plucking these teeth as it goes along, and thereby producing sound. No different from how you would play a guitar or how you would scrape the comb with your finger. And each time the file is scraped results in one pulse of sound or a single syllable. Okay. So that is how the cricket produces its sound. But the interesting thing about cricket sound is that it is highly stereotypic and species specific. Therefore, you can use sound for the identity of the species in most cases. Okay, and that's what makes sound very powerful for ecological studies when we are studying insects like crickets. Even in case of moths, we have studied the bioacoustics of moths, and there are several moth species that have characteristics calls, and not all moth species produce calls. Unlike crickets, in which most species are acoustically active, but moths do produce sound, and this also can be used to identify the species. Bats, this is also something I have worked on, and we have worked on bats of a rainforest, where again we use sound to figure out which species of bat this is. Now, in case of bats, we use the echolocation call. As you know, echolocation calls are ultrasound signals which we can't hear, but you need specialized recorders which can record in ultrasound, and using that, what you will get is different kinds of sounds, echolocation calls, and based on different kinds of echolocation calls, you can tell it is this species. So we tested this reliability of using echolocation calls of bats for species identification, and we know that it is possible to identify bats using echolocation calls. So 90, more than 90% correct classification of bat calls we got, and it is extremely reliable. So what I'm trying to say is sound can be a reliable way of sampling species. Can it be a reliable way of sampling individuals? That is the next level. So in some species, the calls themselves, while the call is species specific, within call there are some features that change between individuals. So now that can be measured to get at the understanding of individual identity. That is possible. Not always, but in many species that is also possible. So at the very least, the presence absence of species, relative abundance, whether in a particular area you have more bats of this particular species, or in some other area there is more bats of this species, all of this can be done with sound without even being present there once you have deployed your recorders, and without culling the animals or killing them or trapping them in any invasive manner. Okay, so that's the power of bioacoustic studies. Now, to that, let us add a little bit of contemporary computational science. We borrow some tools from computer science to understand sound better. And the two terms you must have heard very often these days is artificial intelligence and machine learning. People use it interchangeably, they are not the same. Artificial intelligence is the bigger concept which aims to create intelligent machines which can simulate behavior of animals, of humans. So it aims to do all kinds of things, for example, thinking, sensing, adapting, predicting stuff. Machine learning is a tool which is subsumed under artificial intelligence. Okay, machine learning is used to for artificial AI studies, and it's an application of AI which allows machines to learn from the data without explicitly being programmed to do so. So it's this slight, a subtle difference. Now, for machine learning, what do we need? Of course, there are different stages, but let me take the example of birds. Okay, and I will try to make a case for bioacoustics and for a little bit of machine learning using sound. How do we identify birds? I have put the pictures of six different species of birds. I don't know how many of you can identify these bird species. If you can, then it would be nice if you can write it in the chat box if you're allowed to do that. Which species of birds are this? These six bird species. I shall not tell you which these are, but these are all birds from India. If we have to identify which species it is, how do we identify? One way is that we say, okay, well, these two are similar colored, so they are likely to be more similar. These are more similar to each other color wise. Size wise is another way which you can identify. We when we see, we can identify, but if you ask, how did you identify? That is something that we oftentimes don't think about. Okay, so I want you to think about, how do we identify birds? Take a moment and think, what do you do when you need to identify the bird? Once you have taken a moment, I will tell you how I think I identify bird. Okay, and then we can exchange notes towards the end of this talk. When I am identifying birds, I look at some features. Okay, those features include size. Okay, one very important way of identifying birds is we say whether it is a sparrow plus or a sparrow minus, myna plus myna minus, crow plus crow minus, or a eagle plus eagle minus. Okay, so size. Now say for instance I have spotted this bird. Okay, this bird is definitely bigger than a sparrow, definitely bigger than the myna, but not as large as the crow. Actually, it is crow minus. Okay, so now I know in which category the size of the bird is. Then I look at the shape. For example, I look at the shape of its beak, I look at the shape of its body, I look at colors on the body. If there are different colors in different parts of the body, and I look at certain features. What kind of features? Let's go and examine this. Given the relative size, once you have assessed the size that it is a crow minus, then I go look at the colors. Well, it is a, whether it's black, brown, green, or white, it is, I would say it is brown. If you think of shape, if you look at the shape of the beak, is it long curved, short blunt, short sharp? It is definitely short and curved. Okay, other things, other features that I can ask is, was it found near a tree? Whether it was found near water? Was it on the ground? Well, this one was on the tree. So another feature that I have extracted when I am observing this bird, this is how birders look at birds, right? And if you encounter a new species, these are things you'll keep in mind. I look at certain other features. So there is some barring on the chest, no different color patterns on the body, but on the tail, I can see there are tail bands. If you notice, there are two dark bands of tail. How many? Two. So two tail bands. Okay. Habitat it was found in the tree. And habit, was it found in a group? No, not found in a group. Yes, it was solitary. Okay. Now, once you have done that, then you can put these things together and ask, what is the bird? Okay, I don't know how many of you know this bird. If you do, then good. Otherwise, these are the features: brown, short curved, on tree, two tail bands, and solitary. And based on the answer, the pattern that emerges, it is a raptorial bird and maybe a shikra. It is actually a shikra. Okay, so now let's see, how would we do the same exercise using sound. Okay, bioacoustics, as I said, is a really a new field of science, and therefore, it is still in the developing stage. We are still kind of learning more things, we are still trying to do more. Okay, But I want to use the example of birds to tell you how we can study different bird species. So let's see. This is a for the first one is the Hume's leaf warbler. Dr. Chakrabarti, am I still? Um, can you just prompt if you are being able to hear the sounds? I am. Yeah, I'm playing. No, I'm just playing now. Just a moment.
No, ma'am.
No, no. Oh, that's too bad.
Present, ma'am. Sound.
It is there.
Yes, ma'am.
Okay. Can
Very low, very low.
okay. Can you increase? Yeah, I'll tell you why you could not hear the first two. But red-winged bulbul you could hear, right? I request all participants to increase the volume of their laptops. Okay. Once again, the red-winged bulbul. Okay. Okay. So the reason you could not hear the first one, the people who have managed to hear the first one are the younger people in the audience. Okay, because as we age, our higher frequency hearing ability is lost. Okay, so as you see, if you, this is a spectrogram. On the y-axis is frequency. It is a rather higher frequency call. Okay, it is around between, around 8 to 9 kilohertz. Okay, the most of the energy is around 8 kilohertz. So therefore, even if you will hear it, you will only hear when I jack up the volume very much, the older people. Okay, unless you are old and you still have an excellent ear. If you are somewhat older, above 35, and you can still hear it, that means your ears are really really sharp, so you can congratulate yourself for that. But by and large, this very feeble sound only the very young people will be able to hear. The red-vented bulbul, on the other hand, you see the sound is close to 2 kilohertz, lower frequency, which means these are more deeper sounds, and that these are the kind of sounds older people can still hear. So we have, as we grow old, our high frequency hearing becomes poorer and poorer. So therefore, that is one of the limitations of sampling using doing a service using sound. Okay, if you were hearing the birds, you may have, if you are older, if you are a PI, if you are a scientist, if you are a professor, above 50, very likely you might miss out on hearing some of these birds which are higher in frequency, unless they are very close to you or they are loud. Okay, on the other hand, younger ones, the students will be able to hear these birds. So what do we do now? This is a problem, it's a real problem. Okay, so therefore, the advantage of sound is you can always record it and have a permanent record, and you can visualize this, you can see the sound. And that is what we do. If we do not rely on manual surveys anymore, we do not, back in the days when I was doing my master's thesis, I went to the Western Ghats, and we were doing point counts where we were listening to sounds of the birds and we were making notes of which all species we have found. Okay, also looking at visual, so we were combining both visual and acoustic sampling in the point counts. But this is an error prone technique, and technology has allowed us to improve our sampling by recording these sounds, looking at the sound and identifying. So here I put the bird picture, but now if you have a library of sounds, then you can tell exactly which species of bird it is. In fact, those of you who do a lot of birding, you don't need anything more than the sound oftentimes to tell that, oh, that is this species. Okay, I'll just go ahead. Here, for the lack of time, I have not done this quiz, but it would have been really fun to see. This is a spectrogram. Okay. Now, when you see, it's actually a lot of different kinds of species that I have identified. But forget about this for now. Just pay attention, close your eyes and pay attention to the sound. Okay. That is the sound that we have recorded from a habitat. I'll play it one more time. If you were to look at it, if you were surveying it, you may have heard the cricket. You may have definitely heard the koel, okay, but you may have missed the crow. You may have missed the barbet. The barbet sound is right at the bottom. It is a rolling sound. It's something like that. So you will only hear the barbet or pay attention to the barbet towards the end, because there are some other sounds which are far more conspicuous. So your brain will pay attention to that. This is the problem, not problem. This is how we perceive sound. And therefore, during surveys, oftentimes we miss out on species simply because we have selective attention. We're selectively paying attention to some species. Once again, now see if you can hear the barbet from the very beginning. Barbet has started. Okay, the barbet was there all along. It's just that oftentimes, because it is in the background, you may not actually pay attention to. So these are some of the things that I think that bioacoustics really helps us when we make permanent records of our samples. So if you look at traditional methods where you are going through, dense forests, or you are in an aquatic habitat, or the forest, the grass, say for instance you were working in Kaziranga, then you might actually have to sample from a boat. You may not be able to walk on foot simply because it's too dangerous to walk on foot. Okay, so for instance, uh, traditional techniques where you are on a boat and you are sampling the birds, you're trying to see them. How often are you going to miss the birds? You are going to miss the birds a lot, right? Because of problems of accessibility. And most of the places which are species rich do not have too much access. Okay, a rainforest, or where you have to look at the birds for a very long time, all of these problems are real problems. If you have to sample the rainforest in the canopy, something which I had to do, the problem was just to access the canopy. So you have to climb up into the canopy and then make your recordings. It's very hard. So these are the difficulties of manual survey: the dangerous terrain, time and effort intensive, inaccessible locations, long term monitoring is difficult due to funding. And therefore, bioacoustics can help you in each of these things. Now, if you think of what are the objectives of machine learning algorithms, I'm just taking example of bioacoustics. Machine learning can be applied for many different things. The first step is you have to train with a training data set. Define the features. Remember the kind of features that we talked about in the birds. You define the features, and then based on this, the algorithm converts the data that you have provided into a mathematical representation that define these characteristics. And these characteristics, now you may have some basic features, but the algorithm may come up with thousands, 10,000 of features, okay, which will help the algorithm learn a certain function to be able to predict. Okay, so that prediction is the outcome of the machine learning exercise. And what kind of problems can we solve using machine learning? Not only classification, where we tag the data with appropriate label, that this is a crow, this is a shikra, but also prediction. And here prediction, I am talking about what the patterns of species diversity is right now, how it will be to be in the future. But also clustering. Okay, it is segregating based on similarity, but it is not labeling. Okay, so these are various things that machine learning can do, and I will show you just some examples. For example, classification. All zoology students know this, that whether it has four legs, well, no, and yes. So you, this is a feature which we use to classify fish apart from all other animals. Does it breathe through skin? So yes for frogs, but no for crows and mammals. Does it lay eggs? Yes, no, so on and so forth. This is a classification scheme. Now using sound, we do something exactly the same. So whether it has a certain feature, yes, no. Then you see whether it has some other feature, yes, no. And you keep going. This is a binary classification. This is one of the most simplest form of classification. Using different features, you can come up with how many different kinds of, say for instance, sound types are there. Okay. Prediction, on the other hand, you can have where different species of birds may have different frequency features. Okay. And so what, now once you have this recording, your recording you have taken, you have analyzed the recordings, and you know that a particular species has a particular sound frequency. What can we do with this? You have this call library, and this library of acoustic features. The prediction that can be done, I'm talking about a very simple, it's not a machine learning prediction, I'm just giving you example of what predictions are. Once you have the body size and frequency relationship. Okay, we were talking about a mathematical representation which can be used for prediction, right? So just take this example of this correlation. Okay, so you have a certain relationship between these two, and then when you hear a particular sound of a particular frequency, you can predict what the body size must be. Okay, so this is a form of prediction. Okay, so this can be done. Clustering, you have different kinds of sounds. Okay, and you cluster them based on whether they are similar or dissimilar to each other. Okay, this is another form. So classification, prediction, clustering, all three can be done. Here I have shown you calls of the species called jungle babbler, one of the primary model systems on which we work. And we have clustered this is supervised clustering. You can also do unsupervised clustering of different kinds of calls which are produced in different behavioral contexts. Okay, now, coming towards the end of how we can utilize bioacoustics to study nature, we what we call as soundscape ecology is basically studying the sound of nature to understand the ecological processes and community composition. But we can also use bioacoustics to do phenological studies, to study nature's calendar, if you like. Bioacoustics can also be used for wildlife conservation, okay, to detect rare or endangered animals or to detect threat to wildlife. So, quickly running through the examples, you make recordings, and from the recordings, using the classification, prediction, and clustering scheme, you can tell whether it is biophony, whether it is produced by biological organisms, or is it by the habitat, so geophony, or is it produced by humans, anthropophony. Now, using this soundscape, we can subject it to, typically people use convolutional neural networks. I'll not get into all of these things. And then you can ask various kinds of questions. How many species are present in this area? Which species are abundant? Which are rare? Which area has more number of species? How a species is distributed across a landscape? How climate change is altering species ranges? So on and so forth. Coming back to our birds, how many species are there? With the sound, you can tell, okay, these are the species of birds that were there in this soundscape. Now you can ask, which species are common or rare? Just by size, I'm indicating the ones that are more abundant. I have made them bigger in size, just a representation. So, using the soundscape, you can tell which species sounds are more frequently encountered and which species sound are much rarer to encounter. Okay, so that is a proxy for how common or rare a species is. Okay, species range maps, this is just an example. This is not the result of a bioacoustic survey, but typically if you have bioacoustics recorder across a certain area, you will be able to detect the presence absence of species, and you can come up with range maps for the species. You can tell not just range map exactly at what time of the year that species is detected in that area, whether it is a resident species present all year round, whether it comes only during the summer, comes only during the winter. Even we can tell which time of the day a particular species is found, or even a particular behavior is found. So you can look at all of these things, phenological studies using bioacoustics. Last but not the least, like I said, for conservation, real time data on animal movement, not just species level, individual level. People are using, these are not hypothetical scenarios, these are the uses of bioacoustics. People are using all of these to detect the presence of animals in different habitats, whether they have moved from a habitat or not, whether this habitat is being used, detection of endangered or rare species, and also detection of human sounds. So people have installed sound recorders in the forest to detect presence of gunshots or trees being cut. Okay, all of this bioacoustics is empowering studies in ecology. So all I have to say, this is my last slide to you, just to keep in time. It is a sound way of life, and there are many ways in which technology can empower us, but we need to choose how to use this. When we are using recorders, there's also a problem of privacy concern, something we usually don't worry about in a country like India. But when you record sounds, you're recording everything. You're recording conversations of people. If you install recorders in public parks, you will be basically you are spying on people's conversations, right? So these are things we need to worry about. People are actually worrying about this and coming up with algorithms that actually cut out human conversations and get everything else, so on and so forth. Okay. And since I hope that there are several young students here, and all I want to say that we are done and dusted. We have very little to contribute now, so the future of the earth can only be decided by the young people. So you need to think how you can contribute to ecology. And just to quote Jane Goodall, what you do makes a difference, but you have to decide what kind of difference you want to make. So thank you everybody for listening to me. And I am Manjari. This is ISER Mohali campus. You can see the Shivaliks at the backdrop. I would like to thank all my lab members, and I would like to thank various funding agencies that have funded our lab and our research. And I would give my sincere thanks to Zoological Society, Kolkata, for inviting me. Thank you very much. O- Okay. So first of all, I just want to quickly tell many of you have got the species right. Yes, purple sunbird, red-whiskered bulbul. We also had the flycatcher, the paradise flycatcher female, then rose ring parakeet female, oriole, and the fantail. I think nobody got the fantail, so these were the birds. So very nice to know many of you, Niloy, Subhashri. I see that you guys have got the birds right. So thank you for that. As for the reference material, will it work if I send you a list of books that the interested researchers and students can refer to? There are some books in bioacoustics which are